Namaste to all students. Kushro Pitia, myself Jayoti and Dr. Kushbu Verma from Faculty of Agriculture and Veterinary Science. Today we are going to discuss about Golgi apparatus structure and Golgi body function. And this session is related to cell biology course of program BTech FBT and BSc FST second sem. So before starting the Golgi apparatus structure and the function, you have to understand about the definition of the Golgi apparatus. So Golgi apparatus is an organelle in eukaryotic organism that moves molecule from the endoplasmic reticulum to their destination. Means endoplasmic reticulum, either it is a rough endoplasmic reticulum or smooth endoplasmic reticulum, they will package their material, they will transport that packaged material through the vesicles, and these vesicles will come towards the Golgi apparatus, and further they will be fused with the Golgi apparatus, and then other folding and the modification if required, so it will be happen. And after the completion of all the modification or glycosylation, those packaged material will further go or transfer to their final destination. So the organelle also modifies the product of the endoplasmic reticulum to their final form. So Golgi apparatus is having dual nature, dual role, means they will transfer the material and they will have the modification process for the product of endoplasmic reticulum to give their final form. So the Golgi apparatus is comprised of a series of flattened sac that extend from the endoplasmic reticulum. Now we can discuss about the overview of the Golgi apparatus. So the main function of the Golgi apparatus is the ability to deliver vesicles or packets of various cell products to different locations throughout the cell. And the Golgi also has important function in tagging vesicles with proteins and sugar molecules, which serves as identifiers for the vesicles so they can be delivered to the proper target. The organelle is also called the Golgi complex or Golgi body. Typically, proteins and cellular products are manufactured in the endoplasmic reticulum. The rough endoplasmic reticulum has a number of ribosomes which assemble proteins from instructions contained in messenger RNA. Throughout the rest of the endoplasmic reticulum, these proteins products are folded and further modified. As they reach to the Golgi apparatus, more modification are made, means glycosylation will be happen, and finally the products are packaged within the vesicles, which are labeled by other proteins and the molecule, and the vesicles are further released from Golgi apparatus, and based on their tags or label, they are carried to the appropriate location within the cell by cytoskeleton molecules. So this is the overview of the Golgi apparatus, that how it works. And now come to the structure of the Golgi apparatus. So the structure of the Golgi apparatus is having cis phase and the trans phase. So the cis phase of the organelle is closer to the endoplasmic reticulum, and the trans phase is the side furthest from the nucleus, which secretes the vesicles to various parts of the cells. Means trans phase is the mature phase and cis phase is the forming or budding region of the Golgi apparatus. Further, there are a number of lumen and cisternae through which product flow, and these appears a series of flattened stack on each other, much like the endoplasmic reticulum. Here you can understand the structure of Golgi apparatus by the diagram. So here I am showing the diagram of Golgi apparatus. So by which you can understand more about the structure of Golgi apparatus. So this is the Golgi apparatus. This is forming phase, and these are Golgi complex cisternae, and these are the vesicles. These are secretory granules which are having secretory proteins, and these are the cisternae, and these are the sac or vacuoles. And this one is the forming phase, and this one is the mature or trans phase. Now come to the location of the Golgi apparatus, so where it will be present within the cell. So the Golgi apparatus is situated in between the endoplasmic reticulum and the cell membrane, because it is a kind of a mediator which circulate the material which has been prepared into the endoplasmic reticulum. They will transfer that material to the different locations and mostly to the cell membrane. And most often, the Golgi apparatus appears to be the extension of the endoplasmic reticulum, which is slightly smaller and smoother in the appearance. However, the Golgi apparatus can be easily mistaken for a smooth endoplasmic reticulum. So there is a slightly difference in between the smooth endoplasmic reticulum and the Golgi apparatus. Although they look similar, the Golgi is an independent organelle which has different functions. And if you're talking about the Golgi apparatus presence in the plant cell, so while primarily discussions on the operation of the Golgi apparatus within the animal cells, plant cells also have a Golgi apparatus. In fact, plant cells may contain 100 of these organelles. So it means Golgi apparatus will be present in animal cells as well as in the plant cell. And if you are talking about the key points related to the structure of Golgi apparatus, so there are some questions like in which one of the body fluid is potassium concentration higher than that of the sodium ion? Plasma, perilymph, endolymph, cerebrospinal fluid, or Golgi apparatus? So you have to give the answer, and some other questions also can be asked for checking the understanding level of the student. So if a product is made that need no further modification in the Golgi apparatus, where does it go after leaving the endoplasmic reticulum? It will go to the Golgi body, or it will transfer directly to the destination? And next question can be asked like, Golgi body is made up of single membrane or it is double membranous structure? So these are some key points and some important questions related to the this topic and this session. And now we are going to discuss about the function of the Golgi apparatus. So Golgi apparatus has many functions, but all functions are associated with moving molecules from the endoplasmic reticulum to their final destination and modifying certain products along the way. The multiple sac of the Golgi serves as different chambers for chemical reactions as the product of the endoplasmic reticulum move through the Golgi apparatus. They are continuously transferred into new environment, and the reaction that can take place are different. In this way, a product can be given modifications and or multiple products can be combined in form of large macromolecules. The many sac and folds of the Golgi apparatus allow for many reactions to take place at the same time, increasing the speed at which an organism can produce products. One important function related to the Golgi apparatus is tagging the cellular products. Means they will tag the final product form that where it has to go. So regardless of the product, the vesicle containing the product move from the endoplasmic reticulum and then into the cis phase of the Golgi apparatus. In the layman terms, this is the side facing to the endoplasmic reticulum. The side furthest from the endoplasmic reticulum is known as trans phase of the Golgi apparatus, and this is where products are heated. After having any modification or addition to these structures, the product are packaged into vesicle and then further tagged with markers that indicate where the vesicle need to end up or what is the destination of this product. These tags will be specific for each destination, and these tags can be molecule such as phosphate groups or special proteins on the surface of the vesicle. Once tagged, the vesicle is excreted from the Golgi apparatus on its way to its final destination. So from all the time, tagging will be perfect and tagging will be specific for each product. So this is the main role of Golgi apparatus, that it tag the cellular product for the specific destination. So that's why Golgi apparatus is very much important for the transportation of cellular products or secretory proteins or integral proteins. And one more important function of the Golgi apparatus, which is finalizing the cellular products. So there are many products that are produced by the eukaryotic cells from protein that can carry out chemical reactions to lipid molecules and that can build new cellular membrane. Some products are meant for the endoplasmic reticulum or the Golgi apparatus itself and travel in the opposite direction of the most vesicles. While the endoplasmic reticulum produces most of the products and bases used, it is the Golgi apparatus that is responsible for the final presentation and the assembly of the product. So this is the main function, and often the environment must be slightly different from the present in the endoplasmic reticulum to obtain certain end products. The many sac of Golgi apparatus function to provide many different areas in which reaction can take place in the most favorable of conditions. And in secretory cells or cells which produces large amount of a substance that your body need, the Golgi apparatus will be very large. Means Golgi apparatus will have more work, basically more work to distribute the final products to their final destination. So that's why Golgi apparatus will be large in these kind of a cells which are having a nature of secretory. And consider the cell in your stomach that secrete the acid. This is the best example to understand the role of the Golgi apparatus, because Golgi apparatus will be large in these kind of a cells and the acid is produced by the reaction in endoplasmic reticulum and is modified as it goes through the Golgi apparatus. Once to the trans side of the Golgi apparatus, the acid is packaged in a vesicle and sent towards the cell surface. As the vesicle joins the plasma membrane, the acid is released into the stomach so it can digest your food. So this is the best example by which you can understand the role of the Golgi apparatus. Now come to the theory behind the Golgi apparatus function. So you have to understand this theory. So the most prevalent theory of how the Golgi apparatus forms is the cisternal maturation model. This model suggests that sac themselves tend to move from cyst phase to trans phase of the Golgi apparatus over time. New sac are formed closer to the endoplasmic reticulum. These sac age as they move towards the trans phase of the Golgi apparatus and their products become fully mature. Now come to the specific products which have been modified by the Golgi apparatus. So it may seem like there could never be enough lipids to produce the continual flow of the cell's membrane needed to continually make transport vesicles between the endoplasmic reticulum and the Golgi apparatus. However, there are constantly segments of the cell membrane being produced and recycled by the endoplasmic reticulum. Golgi apparatus, lysosomes, and other organelles in the cell, as well as the outer cell membrane itself. The Golgi apparatus and endoplasmic reticulum work together to produce new cell membrane as well as recycle the cell membrane of vesicle by merging two membranes when vesicles are absorbed. The Golgi also creates lysosomes. So here you can understand that from where lysosome, which are suicidal bags for the self and suicidal bags for the cell, and if any kind of a damaged material present within the cell, so they can digest and they can degrade that dead material. So lysosomes are created by the Golgi apparatus and these sac contain digestive material. The sac are pinched or from the Golgi apparatus, and they are used to process the material which have been phagocytized or to digest organelles which are no longer function, means they are dead. So lysosome delivers raw ingredients to the endoplasmic reticulum for further modifications. Now come to the Golgi apparatus function in plant cells, because in animal cell there kind of a various proteins, secretory proteins or integral proteins or some acid they produce. So there are a lot of work for the Golgi apparatus in animal cell. But if you are talking about the function of the Golgi apparatus in the plant cell, so within the plant cell, the Golgi apparatus serves the additional function of synthesizing the major polysaccharide molecule which help into the formation of the cell wall. To do this, plants often have many more Golgi bodies than an animal cell. Further, plant cells do not contain lysosomes. So, lysosomes are basically present only in animal cells. They are absent in the plant cell, and these digestive organelles are replaced in the plant with the central vacuoles, because central vacuoles are having similar nature uh, as the uh, lysosome. They also degrade the dead material, and which serves a large lysosome as well as an organelle to store the water. Thus many vesicles from the Golgi bodies of the plant move to the vacuole and fuse their content with this large organelle. So here in the plant cell, vacuole, central vacuole behave like a lysosome, and generally lysosomes are absent in the plant cell. So this is the sum up of this session, and if you are talking about the key points or key questions related to the this session, so you can simply understand by this, like Golgi apparatus only present in the plant cell. This is true or false? And second question is, Golgi body apparatus is not responsible for the synthesis of proteoglycan. So this is true or false? So these are some key points related to this session, and this session is now sum up, and this session is powered by digital version 2.0 Jyoti Vidyapeeth Women's University Jaipur. If you have any query, then mention in the comment box. I will resolve it in. Thank you.